Yeah, good afternoon, students. Welcome back to Botany second year's class. So yesterday I have started with the second LAQ, that is from the 11th chapter, biotechnology, principles of biotechnology. I have started that LAQ, but I could not able to finish it off properly. So even I didn't felt that I am happy with that answer. So we'll go along again that question along with the second LAQ also from that chapter, because there is a link between these two LAQs. If you know one LAQ, at least you can, if that was not given, second LAQ you can answer where you can easily get 4 marks. It's not a big deal to get 4 marks from that. But to answer that, you should have the use your intelligence, that is the somewhat knackly, you have to neatly write the answer, that much. So the first one in this is the process of rDNA technology. So in this, what we have to say, in the process of rDNA technology, we have to say different steps we have to say. So the first one is isolation of desired gene. Otherwise, we will say isolation of DNA. So in the isolation of DNA step, what we will say, isolation means separation, taking out. So in this, the desired DNA also comes, as well as the host DNA and the vector DNA. All three will come over there. Okay. So in this, first of all, we have to take the cell from where we have to separate the genetic material, and that cell, first of all, need to be insect free, that is the microorganisms free, that need to be sterilized first with the different cells are need to be sterilized in different ways. The cell wall, the first of all, the plant cells need to be sterilized, plant cells and the animal cells, fungal cells, as well as the animals, and okay, and the viral organisms. All these ones has to be sterilized. Bacterial cells need to be sterilized first. Bacterial cells, and after the sterilization, that means once they are dipped in the concentrated soap water, then all the microorganisms will be removed from the outside body. Then the cells need to be immersed in the different chemicals, so that enzymes, so that the cell wall will be degenerated. Okay. So plant cells, cellulase is the enzyme which is going to be used, and the for the animal cells that does not have the cell wall, so no need to discuss about that. And the fungal cell, there is a chitinase enzyme, and for the bacterial cell, it's a lysozyme enzyme. Okay, so like this, 3 different enzymes are needed for the digestion of the cell wall of different organisms. And after that, once the cell wall was digested, then cell wall should be digested. That is the first one. And next one, what we have to do? We have to now with the entirely, it is having the plasma membrane. Then the plasma membrane can be removed, can be digested by the some sort of the concentrated enzymes. So very easily we can do it. And after that, the completely the cytoplasm along with the cell organelles will be present. That is the in the isolation of the DNA. Now comes this one need to be taken. So here we are taking the cell where we have to take the genotype of the organism, and the vector where we have to transfer the recombinant DNA into the host cell as well as the desired DNA from which organism we are taking to make the rDNA technology. That desired gene need to be taken out. That is also need to be isolated like this only. In this one, we have said all the 3 different cells and the different types of the enzymes of how to separate the genetic material. Now, next one will be the second step will be the separation of not DNA, separation of the genetic material. So for this, what we have to do, we have to take the test tube, and in the test tube, the complete cytoplasm and the cell organelles need to be kept, and that need to be does the centrifugation. Once we have done the centrifugation, then the heavier particles will settle down at the bottom of the test tube. After that, these in this one, in the cell, the heavier particles are present in the cell. That is nothing but the genetic material which settles down at the bottom of the test tube, and some materials will be just like floating in the semi-solid liquid-like structure. Okay, so this need to be separated by the elution method. By a copper wire, we can easily separate the DNA fragments, floating DNA fragments. That is called as the elution. And after that, we need to take out we need to do the cutting of the